Hi there. We are going to talk about chapter 4 today on cell structure. So we're going to kind of take a quick look at some of the key points from chapter 4 and um, highlight some of the important things. So I'm not going to go into every single organelle, but we're going to take a look at some of the important organelles and some key concepts from chapter 4. Okay, so one thing that you should notice on your note sheet is cell fractionization. Cell fractionization is a process where scientists study all of the different organelles by size. And what they're doing is they take the cells and they basically put it in a blender and they break apart the cell membrane and then they put it into a centrifuge and they spin it so that it separates out the organelles by size. So they'll put it on a centrifuge at a certain speed, and then that separates out the largest particles. And then they will separate the top portion of it. So you can see here the top portion. They take that and put it into a new tube, and then they spin it again. And then that separates out the next size organelle. And then they take the top portion and spin it again, and then that separates out the next size. So each time they spin it, they're getting different sizes to separate out. And so that's cell fractionization. Once they've separated out all of the different organelles, then they can study and analyze each of those organelles. So this is just showing you that process. So they take a sample of tissues, so a bunch of cells, and they homogenize it or basically put it in a blender and break apart all of the cell membranes, and then they centrifuge it and spin it and separate and spin and separate and spin and separate. Okay, one of the organelles that we are going to take a close look at, and we'll talk even more about this when we talk about chapter 5, is the cell membrane. The job of the cell membrane or the plasma membrane is to be a selective barrier. It defines the cell, so it separates out the self, the inside from the outside, and it also controls what enters and leaves the cell. So it's a selective barrier. Some things can pass through it, but some things cannot pass through it. And in chapter 5, we'll get into more detail about exactly what can and cannot get through it and why it can and cannot get through it, and how we've got active and passive transport. So we'll get to all of that in the next chapter. But for this recording, we're just going to stick to talking about some of the organelles and their important functions. So the plasma membrane is a selective barrier that allows the passage of oxygen, nutrients, waste, and it's also going to determine the volume of the cell, the size of the cell, what's inside of the cell. There are two types of cells. There are prokaryotic cells and there are eukaryotic cells. And there are 3 domains. And of those 3 domains, 2 of them are prokaryotic. And so that's going to be the bacteria and the archaea. They are going to be prokaryotic cells. And then we've got the rest of the kingdoms would fit into the eukaryotic cell type. So protists, fungi, animals, and plants are all going to be eukaryotic cells. So the 3 domains are going to have different types of cells. So bacteria and archaea are going to be the 2 prokaryotic, and then the domain eukarya is going to have the protists, fungi, animals, and plants. And we'll talk more about that a little bit later on in the year when we get to classification. But all cells have some basic features. So all cells are going to have a plasma membrane that surrounds and defines them. They're all going to have a cytosol, which is a semi-fluid substance. So you might think of cytoplasm. Cytosol is going to be kind of the the liquid jelly substance, and then the cytoplasm would be that with the organelles in it. So the fluid substance is called the cytosol. And they're all going to have chromosomes, so they're going to have genes and hereditary information. And they're all going to have ribosomes because they need to make proteins. Proteins are the key to the functionality of all cells. So all cells are going to have a plasma membrane, a cytosol, chromosomes, and a ribosome and ribosomes. Prokaryotic cells are going to be characterized by having no nucleus. So they've got no nuclear membrane that separates the genetic information from the rest of the cell. They do, however, have the DNA kind of clumped together in a region or an area called a nucleoid, but it does not have a membrane around it, so it's not defined as a nucleus. And they also have no membrane-bound organelles, which means there's no mitochondria, there's no chloroplasts, there's no endoplasmic reticulum. None of those membrane-bound organelles are in the cytoplasm of the prokaryotic cells. And then the cytoplasm would be just bound by the plasma membrane. So again, there's no membranes that are separating the different compartments in a prokaryotic cell. And they're typically smaller in size, so 1 to 5 micrometers in diameter, so typically 10 times smaller than most eukaryotic cells. So here's a diagram showing you the bacteria. So a bacteria is an example of a prokaryotic cell. You can see the nucleoid region in here, so that's going to be the bacterial chromosome. A little bit later, we'll talk about plasmids and how they might have extra DNA, but their primary chromosomal DNA is in that nucleoid region. And eukaryotic cells are characterized by having DNA in a nucleus, so the nuclear envelope is going to define the area where the chromosomes are found, the genetic information. And they also have other membrane-bound organelles as well. So they're going to have an endoplasmic reticulum, a Golgi apparatus. They're going to have mitochondria and chloroplasts and vacuoles and lysosomes, all of those different organelles that are membrane bound. And they're generally larger than prokaryotic cells, so typically 10 to 100 micrometers in diameter, so larger than eukaryotic cells. And so we kind of need to look at surface area to volume, and that surface area to volume ratio plays a very important part when we're looking at cells. Cells are typically small because they need to get nutrients into the cell. They need to get oxygen in, they need to get carbon dioxide out, they need to get waste out. And if you have a small surface area to volume ratio, then you're going to, if you have a large surface area to volume ratio, then you're going to be able to get nutrients into the cell. As a cell gets bigger and bigger and bigger, that surface area to volume ratio decreases, and it's harder to get the nutrients all the way through into the cell. So if you look at this diagram here, this little one has a surface area. So calculating and doing all of that math, if you take the surface area of it, you've got a surface area of 6. So each side is 1 unit, and then the length times width times height is going to be the total volume is 1, and so the surface area to volume ratio is going to be 6 divided by 1, or 6. So that's a pretty big number. If you look at the bigger cube over here, which has a longer side, when you do that math, you're getting a 1.2 surface area to volume ratio. So the bigger cube has a smaller surface area to volume ratio. And if you think about getting nutrients from a little side all the way to the middle, things can diffuse or move into that cube much easier if the cube is small. If it is going to have to diffuse all the way to the very middle of it, that's going to take some time. And so it has to move through that membrane to get in there. And so the larger the cell, the longer it is going to take for things to diffuse across that surface all the way into that volume. So if you took that same big cube and you chopped it up into smaller pieces, then each of those smaller pieces are going to be more efficient at getting the nutrients in there. So if you think of let's say a classroom, and let's say you are walking by a classroom and it's a small classroom and you want to pass a note to your friend who's in that classroom. So you walk up to the door and you could pretty much hand them a note or just take a few steps and give them the note. Now imagine that they're in a big gym filled with hundreds of people and they're in the middle of that gym. It's going to take a lot longer for you to pass them a note if you have to walk all the way across the gym to get to that person. So a larger area is going to take a lot longer to get that note in across that membrane. Okay, and we're going to do a lab next week where we're going to take a closer look at surface area to volume ratio that will hopefully visualize it a little bit better for you. Okay, so because eukaryotic cells do have those membranes, they have things compartmentalized or partitioned, and that's going to make those cells more efficient. So having things compartmentalized and having roles of each of those compartments, they can make that cell much more efficient. And plant cells and animals typically have most of the same organelles, with obviously some exceptions. So here is a diagram of a typical animal cell. And not all animal cells are going to have all of these components, because you can see you've got little microvilli here, you've got a flagella. So some of those organelles, it's just showing you those generalized characteristics. We're not going to get into all of those different organelles, but you should be able to recognize all of those. And then here is a typical hypothetical plant cell. And so you can definitely see some differences between a plant cell and an animal cell. Biggest differences are that large central vacuole in a plant cell, the cell wall, the chloroplasts, which an animal cell does not have. And then animal cells will have the centrosomes that help with cell division, which we'll talk more about later. So let's take a quick look at a little video here, BioFlix, to take a tour of an animal cell.
Even if you're just studying, a lot of work is going on in your cells. Let's zoom down and check it out. We've arrived in the space between two cells. A sticky coat called the extracellular matrix holds the cells together. Each cell is surrounded by a flexible plasma membrane with an incredible number of projections, docking stations, and channels. Let's dive into one of these channels to enter a cell. Whoa, look at this place. These girders and cables make up the cytoskeleton, the structural framework of the cell. They also serve as tracks for transporting cargo from one place to another. All this activity in the cell requires energy in the form of ATP molecules, which are made here in the mitochondrion. Notice the outer membrane and the inner membrane with its numerous infoldings. Many of the molecules involved in making ATP are built into the inner membrane. All those folds increase the inner surface area, enabling more ATP to be made. Moving towards the nucleus, we pass by layers of internal membranes. The nucleus is enclosed by a double membrane called the nuclear envelope. Let's enter the nucleus through a pore. The nucleus houses the genetic material of the cell, DNA, which carries the blueprints for making the cell's proteins. Almost 2 meters of DNA is crammed inside the nucleus. How does it all fit? The DNA is wrapped around proteins like thread wrapped around spools. Look, this section of the DNA has unwound, and a different protein has attached to the DNA. DNA is being used as a template to make mRNA. mRNA molecules travel from the nucleus to the cytoplasm, carrying the instructions for making specific proteins. In the cytoplasm, a ribosome clamps onto a strand of mRNA. The ribosome ratchets along the mRNA, building a new protein. Some proteins stay in the cytoplasm. Others, like this one, are processed in special compartments within the cell. Protein processing and certain other metabolic activities occur in the endomembrane system, the cell's network of internal membranes. The endoplasmic reticulum, or ER, is part of the endomembrane system. There are 2 types of ER, rough and smooth. Rough ER is covered with ribosomes. Smooth ER lacks ribosomes. Lipids are made in the smooth ER. Let's go inside the rough ER. Note that you can still see the ribosome on the outside surface. The ribosome is manufacturing a new protein, which continues to grow inside the ER. Completed proteins move to the edge of the rough ER and depart in a vesicle that buds off from the ER membrane. Some vesicles fuse with the Golgi apparatus, another component of the endomembrane system. In the Golgi, proteins undergo further processing. Finished proteins are then packaged in vesicles that pinch off from the Golgi and are transported along cytoskeleton tracks. Some vesicles bind with the plasma membrane, secreting their contents outside the cell. Other vesicles, called lysosomes, contain digestive enzymes. Here, a lysosome fuses with a worn-out mitochondrion and breaks it down. Each of the trillions of cells in your body is a dynamo of activity, requiring millions of ATPs every minute. But most people are unaware of all this activity in their cells.
So that was a plant cell. I'm sorry, an animal cell. Let's take a quick look at a plant cell.
Both animals and plants are made up of cells. Their cells have many features in common, but there are a few significant differences. Let's look inside a leaf to take a closer look at a plant cell. First, we encounter a protective cell wall outside the plasma membrane. The cell wall is made from strong cellulose fibrils. Once inside the plant cell, we see the large central vacuole, which regulates the composition of the cytoplasm, creates the internal pressure that is characteristic of plant cells, and stores various compounds produced by the cell. Plants make their own food by photosynthesis in chloroplasts. Light passes through the 2 membranes of the chloroplast and strikes these green discs, where light energy is converted to chemical energy. The sugar molecules produced by photosynthesis can be made into other molecules or broken down for energy. When sugars produced by photosynthesis are broken down, their energy is used to make ATP in mitochondria. ATP powers the work of the plant cell. Most organelles, like mitochondria, are found in both plant cells and animal cells. So the next time you pass by a plant, remember that we have more in common than meets the eye.
Right. So a nice overview of both the plant and animal cells, and you can see some of those organelles and how they work together. It also talked about the endomembrane system in the first video, the plant cell video. And the endomembrane system is really all about how all those membranes are kind of connected together and work together. So if we look at the nucleus, the nuclear membrane obviously surrounds the nucleus, and it contains the genetic information inside the nucleus. The nuclear membrane is actually connected to the endoplasmic reticulum. And again, we've got the smooth ER and the rough ER, and the rough ER has the ribosomes attached onto it. Proteins are being produced and sent into the rough endoplasmic reticulum. The smooth ER is going to produce lipids and process toxins, but they are kind of all connected together. And then the endoplasmic reticulum kind of is tied to the Golgi apparatus through vesicles. So what will happen is proteins will be produced in the rough ER, and then they get packaged in these little vesicles, which will then get shipped to the Golgi apparatus. And that little vesicle will merge with the Golgi apparatus, and then the proteins that are in them will get processed and packaged, and then they get shipped out to the plasma membrane, where those proteins will either become embedded in the membrane or will be secreted out of the cell. And so secretory proteins will be released through the Golgi apparatus. So they're going to transport it and ship it to the plasma membrane. So the endomembrane system is really all of those membrane-bound organelles that are kind of working together to process things like proteins and lipids. And here's just another diagram of that endomembrane system, but this is talking about more of the proteins. And so when we've got proteins that are embedded in the membrane, and we'll see that more when we talk about chapter 5, but they're going to be produced by those ribosomes, and they're going to then be embedded into the in the membrane of the endoplasmic reticulum, or they're going to be in the endoplasmic reticulum vesicles that are then going to get shipped to the Golgi. And so if a protein is designated for the plasma membrane or the cell membrane, then it's going to actually get embedded. And so you can see this picture up here in the middle. The protein is going to be embedded in the membrane of that vesicle that's going to the that's going from the endoplasmic reticulum to the Golgi apparatus. And then it's going to get packaged, and it's going to eventually make its way into a vesicle that will merge with the cell membrane or the plasma membrane. And so that vesicle, that membrane of the vesicle, will become part of the membrane of the cell. And so whatever protein is embedded in that vesicle membrane is now going to be embedded in the membrane of the protein. And if it's a secretory protein that's going to be secreted out of the cell, then that would be inside the vesicle. And when the vesicle merges with the membrane, then that inside of the vesicle becomes outside of the cell, and then those proteins are released out of the cell. And so that's kind of what this diagram is showing you, is it's showing you the synthesis of those membrane components or those secretory proteins that will get released from the cell. And then if we take a look at the mitochondria and chloroplast real quick, we're not going to get too much into the structure. When we get to our energy unit, we'll definitely talk more about the structure of the mitochondria and the chloroplast. But the mitochondria is producing energy, and the chloroplasts are converting solar energy into chemical energy. But both of those, the mitochondria and the chloroplasts, those are double membraned organelles, and they tie eukaryotic cells to prokaryotic cells when we look at evolution. And we'll get more into the endosymbiotic theory when we get to evolution. But both of them have similarities with bacteria. And because they are very similar to bacteria, that is evidence towards the endosymbiotic theory. They are both enveloped by a double membrane system. They contain free ribosomes and circular DNA molecules. So they have their own genetic information. They have their own DNA. And because they have their own DNA, that supports the endosymbiotic theory, which we'll take a look at in just a minute. But they grow and they reproduce somewhat independently in the cell. So again, more support for that endosymbiotic theory. And so what the endosymbiotic theory is that what is now the mitochondria and the chloroplasts, those were originally prokaryotic cells that got engulfed by other prokaryotic cells and then started working together in a symbiotic manner to then become eventually the eukaryotic cells that we now know of as eukaryotic cells today. So if we look at this picture here, so in the endosymbiotic theory, what we're looking at is in early evolution, the first cells that evolved were prokaryotic cells. And then what happened is the cell membrane started infolding upon itself, and some of that membrane material formed the nuclear envelope and things like the endoplasmic reticulum and Golgi apparatus. So kind of an early prokaryotic cell forming that first nuclear structure. And then that endosymbiotic theory where one of those then engulfed a cell that could do cellular respiration. And so now that becomes that prokaryotic cell, that mitochondria, early mitochondria, is now producing energy for that larger cell. So a symbiotic relationship with that mitochondria inside of another cell. And then some of those also engulfed photosynthetic prokaryotic cells, and those evolved into chloroplasts. And so the mitochondria and the chloroplasts now in a photosynthetic eukaryotic cell, which then evolved into things like plant cells. And so that's the endosymbiotic theory. First, the infolding of the cell membrane, forming the nuclear envelope and those other membrane-bound organelles, then engulfing prokaryotic cells that could do cellular respiration and then working together, producing energy now for that new cell, and then other cells engulfing photosynthetic prokaryotic cells that then became the chloroplasts. Okay. Also in chapter 4, we're looking a little bit at things like microtubules, microfilaments, and other intermediate filaments. They all give the cell its structural shape. So the cytoskeleton of the structure, we're not going to get into all of the details, but just know that there are microfilaments and microtubules that help with maintaining the shape of the cell. They help to move things in the cell. They help with things like muscle contraction. So those are going to, those filaments and fibers give the shape and the structure within the cell, especially when we're talking about animal cells, that don't have a cell wall. So animal cells are going to have that cytoskeleton. And plant cells, of course, things that distinguish a plant cell from an animal cell is the cell wall of the plants. And so animal cells do not have that cell wall, and it is going to be made up of mostly cellulose microfibrils that produce that cell wall, and it helps to protect the plant cell and maintain its shape. Look at this video clip.
Animal cells in multicellular tissues are usually joined by tight junctions, desmosomes, also called anchoring junctions, and gap junctions, also called communicating junctions. Plant cells are often joined by plasmodesmata. Each type of junction has specialized functions, such as preventing fluid leakage between membranes or allowing the free passage of small molecules and ions between adjacent cells. Tight junctions, which bind cells together, are found predominantly in epithelial tissues, such as the lining of the intestines. Tight junctions form a barrier that prevents fluids from moving between the intestinal space and the space between cells. Desmosomes, or anchoring junctions, appear as thickened patches in the cell membrane region between two cells. Desmosomes contain specialized proteins, such as keratin, the same protein found in fingernails and hair, that increase the rigidity of tissues. Desmosomes, such as those found in epithelium, smooth muscle, and many other animal tissues, are button-like junctions that bind cells together and also function as anchors for fibers in the cytoskeleton. In gap junctions, or communicating junctions, two cells are separated by a small gap, which is bridged by specialized channels that allow the passage of water and small molecules. Gap junctions help coordinate the activities of adjacent cells. For example, a hormone that stimulates one cell will often activate adjacent cells as well by the passage of intracellular signals through gap junctions. Plant cells, unlike animal cells, are surrounded by thick walls that form rigid tissues. They do not need the junctions found in animal tissues. But some higher plant cells are interconnected by plasmodesmata, tube-like structures that penetrate through cell walls and form fluid connections between adjacent cells. Like gap junctions in animal cells, plasmodesmata allow the free exchange of small molecules and help coordinate the activities of neighboring cells.
And when we get to our cell communication unit, we'll talk a little bit more about those connections that allow molecules to move from one cell to the next and send signals and communicate with each other. Okay, and then we've got finally the extracellular matrix of an animal cell. And so again, the animal cell doesn't have that rigid cell wall to hold the structure together. So there are also extracellular components that kind of help with cell communication, cell signaling, receiving messages. And so there are fibers and webs and things that are also on the outside of the cell that give the cell some support and structure as well. Okay, we're going to stop there. And then we will do another video with chapter 5 talking about membranes and transports. So I'm going to stop sharing right now, and we will pick up with another video with chapter 5. All right, that's it for today.